Good morning, everybody, and welcome to today's session. I'd like to talk to you guys today about some of the advances that have been made in technologies in confocal microscopy and how that's driving imaging like we've never seen before. So some of the topics that I'd like to cover this morning include high sensitivity spectral imaging, live cell imaging, high-speed imaging, and lastly, macro to micro imaging. So starting off with high sensitivity spectral imaging, when you think about your traditional confocal microscope system, typically what you've been presented with is two choices, either a spectral-based detection system or a filter-based detection system. So filter-based detection systems, they use predefined bandpass filters and dichroic mirrors to select the detected wavelengths of emitted light. And what this typically is associated with is a higher transmission efficiency. So therefore, you can get a stronger detection signal from your sample. But some of the drawbacks of a filter-based system is that you have a limited range and combination of fluorophores that can be detected. Now, on the other hand, if you have a spectral-based detection system, it allows the users to define the wavelengths that they'd like to detect. And this allows for a flexible use and combination of fluorophores, but it's typically been associated with a lower light transmission efficiency due to loss of light from diffraction gratings and reflections in the light path. Now, the advances that have recently been made in spectral detection include the true spectral detection concept, which is featured on the brand new FlowView 3000 system. And what it allows is it allows for up to 4 channels of all true spectral detection. We use a patented volume phase hologram transmission light path. And so what this allows is for a higher transmission of light from the emission to the detector. It gives the users nanometer precision bandwidth definition, all the way from 1 nanometer up to 100 nanometers. And it gives full flexibility from 400 to 800 nanometers for each channel. And so with this new technology, multicolor imaging is now made easier. You can get clear spectral separation from your sample, allowing for multiple stainings and flexible spectral bandwidth, and you can optimize for your emission collection. You can detect more fluorophores than the number of physical channels, up to 16 channels with only 4 detectors. You can remove autofluorescence background, and it allows for a higher throughput by precisely positioning the detection bandwidth relative to the laser line. So this modern spectral imaging widens your possibilities and allows you to image more colors more accurately.
Was that image taken with an FV3000?
This image was indeed taken with an FV3000. This was a rainbow sample imaged with spectral imaging and unmixed to allow for identification of visible Purkinje cells, dendrites, and axons, as well as some aspecific stainings. With our true spectral detection system, we can also do live unmixing. So if you have fluorophores that are very close in their emission wavelengths, you can unmix your sample to more accurately define where your proteins are located within the cell. So for example, in this image here, we have live blind unmixing occurring of endosomes in blue, M-ametrine in the plasma membrane, the nucleus in orange, as well as actin in purple. So with this detection system, there are 2 modes that you can use. There's a variable bandpass filter mode, which allows you to simultaneously image both bright and dim fluorescent signals within your target without saturating the signal. So, invariably, whenever you have multiple fluorophores in your sample, they're not all going to be emitting light at the same intensity. So you want to be able to adjust your detectors in order to make up for that brightness or dimness. So you can adjust the sensitivity of each channel individually so that you're equally capturing your fluorescent signals. In its second mode, what you can do is do a lambda scan, where you scan through a specific spectrum in specified steps. This enables you to acquire a spectral image with high wavelength resolution, as well as you can adjust for the sensitivity of bright and dim colors here. So you can more accurately step through the spectral emission of your sample to capture accurately the locations of the fluorophores, especially for fluorophores that are closely overlapping. You can very accurately unmix those samples to more accurately detect your samples. So the next thing that we're going to talk about are the advances that have been made to enable you to image live cells with a confocal microscope. And in order to image live cells, you really have to optimize all components in your system, not just one of them. So from excitation in terms of your scanner and its scan speed, you want to be able to optimize speed to minimize phototoxicity. In terms of detection, you want to have not only flexibility through spectral detection, but you also want to maintain sensitivity so that you don't have to overstimulate your sample with the laser power. In terms of optics and the microscope, in terms of live cell imaging, you want to be able to match the refractive index of your sample through silicone immersion optics. And for long-term imaging, Z-drift compensation with autofocus to really maintain focus on your sample. And as well for live cell imaging, it's important to maintain environmental control to really capture the true physiological reaction of your sample. So in order to maintain sensitivity in your system, one of the ways in which you can do that is to use high sensitivity detectors. Commonly used for this application are GASP detectors, which have a very high quantum efficiency throughout the visible range, up to 750 nanometers, as well as you can use hand-selected multi-alkali PMTs to provide flexibility out to 800 nanometers of detection. Now for time-lapse imaging, you can achieve higher quality live cell data with more physiological relevance by really optimizing sensitivity, flexibility, and speed. In this example here, we have NK cell-mediated cell killing after therapeutic antibody application in blue. And so the GFP-labeled NK cells in green, and DAPI uptake is marking the dead cells. So you can really capture those high critical physiological events over a long period of time without damaging your cells. Now, another important consideration for live cell imaging is the type of optics that you're using. Typically, what's associated with live cell imaging are either oil immersion optics or water immersion optics, but neither of these match the true refractive index of your live cell sample. But right in the middle here, if you use silicone immersion optics, you're going to be matching the refractive index of your sample. And why that's important is the fact that a refractive index mismatch is going to lead to lost light due to the fact that the focus is incorrect due to the point spread deformation of your sample. So on the surface of your sample, if you have a refractive index mismatch, it's not really going to lead to that much aberration. But at the further, deeper that you go into your sample, you're going to be missing out on focus in your sample, which is going to lead to less light, as well as decreased resolution in your axial direction. So when you're able to use silicone immersion objectives here, you're able to capture the true reality of the axial projection of your image here. So if you have a mismatched refractive index, your resolution in Z is going to be reduced from the elongated point spread function. And what that's really going to do is it's going to decrease your axial resolution. So both of these images were acquired with the optimum Z setting for the lens, but you can see the profound difference that imaging with a refractive index matched to the sample really makes. You can get a perfect 3D reconstruction in large living organisms. So as seen here, you have a zebrafish eye that was expressing the cell cycle indicator Fuji. And the silicone immersion optics have allowed us to accurately reconstruct the zebrafish eye here and allow the user to capture high resolution images in large living organisms. So moving on to high-speed imaging. High-speed imaging is critical to allow for the user to reduce the photo-bleaching and phototoxicity in a sample, and it allows you to more accurately image in vivo samples here. So this image of a zebrafish here was captured using a macro 2x lens and resonance scanning, and you can really see that live real-time physiological events that are occurring within the whole organism itself. Now, typically, the use of a resonance scanner has meant that there is a trade-off between the field of view and the speed at which you can capture. But on the resonance scanner featured on the FlowView 3000, you can capture up to 30 frames per second at a full field of view. And by clipping down to 512 by 32, you can gain up to 438 frames per second, which is really going to allow you to capture those really high-speed physiological events such as calcium imaging, as well as others. So it's important to maintain that field of view so that you can capture your physiological data in the broader context of what's going on in your sample. So in this example here, using the resonance scanner, we're able to see that platelets bound to a thrombosis in blood vessel of a mouse. You can see the lifetime events that are occurring. And this image itself was taken at a full field of view at 30 frames per second. And so not only can you see this area here where the action is occurring, but you can also see it in the wider context of the sample itself. So the reason why resonance scanning offers such an advantage for live cell imaging is that it allows you to reduce photobleaching and phototoxicity. The triplet dark state is when you have overexcitation of your fluorophore, which leads to a dark state, and it has a much longer lifetime than your typical fluorescence, which contributes to the photobleaching that you typically see when you overexpose your sample. Now, a normal galvo scanner is going to be scanning in the 2 microsecond scale, possibly longer, depending on your dwell time. Whereas the resonance scanner reduces this photon flux with a scan speed of around less than 0.1 microsecond per pixel. So very fast, and even when you average resonance scans to improve your resolution, you can reduce photobleaching as well as reduce reactive oxygen species in your sample. So in this example here, what we have is a cultured neuron, and it was captured using resonance scanning time-lapse with a 100x silicone objective. So again, we're matching that refractive index of our sample in order to maintain our resolution in Z. It was deconvolved using CellSense, and this frame was averaged used 7 times. And even averaging 10 times with the resonance scanner, you can produce a frame rate of less than 1 frame per second compared to 1 frame per second for 2 microsecond pixel galvo scanning at the same field of view. So even 7 times averaging, you're about twice as fast as you are with typical galvo scanning and maintaining happy cells. So lastly, we'd like to talk about some of the advances in macro to micro imaging and confocal microscopy. And this is really going to allow the user to capture the entire range or the entire image of your sample and dig down into really fine details. And so with the new optical light path in the FlowView 3000, you can acquire images using 1.25x objectives all the way up to 150x objectives. So with this concept, you can acquire the whole tissue image and then focus in on the regions that you'd really like to acquire fine details. So for example, this image on the left here was acquired with a 1.25x lens, and you can clip down into images and get the details and the resolution that you're looking for here. This image itself was captured in one shot with, again, a 1.25x lens. And so these large overview images then allow you to see areas of interest that maybe you want to focus in on and image with a higher magnification. So you can combine these large images and high resolution in a really efficient way. And one of the advantages of this is it's really going to speed up the rate at which you can image your entire sample. So even if your sample is too large to capture in one shot, you can stitch it together to seamlessly create a field of view where you can image your whole organism. So this image here was captured with 2x2 tiling and stitching. And with this method, you can motorize and automate multi-area imaging to cover images that exceed a single field of view. So some of the common needs in biology today that can use macro to micro imaging. For cell biology, in terms of microimaging, you can resolve superfine structures down to 120 nanometers resolution, whereas on the flip side, on the macro side, you can get whole small organism imaging and whole slide imaging. In terms of cancer biology, you can count small particles and get high resolution and segmentation analysis. And then on the flip side, you can turn around and see the entire tumor in the context of larger tissue. And lastly, in terms of stem cell biology, what this technology enables you to do is you can dig down and visualize the stem cell markers at the cellular level, but then you can turn around and you can image the entire colony or tissue in one shot. And so what this means is that confocal microscopy as it stands today is allowing you to image samples that's typically only been reserved for other imaging methods. So at this point, I'd like to take any questions.